Hi everyone, thanks for staying for the last talk of the day. My name is Cristiano, I'm a PhD student, about to get done. I got 3 supervisors. One is Dr. Glaucia Maria Pastori, my co-supervisor is Dr. Francisco Barros, and I got a third supervisor, which is Dr. Paula Jaureggi, during my split PhD. I'm from Brazil, and I'm going to talk, my talk is about application of biological product. The title is Surface Activity of Surfactin Under Extreme Conditions, pH, temperature, and ionic strength. A prospection for microbial enhanced oil recovery. So basically I'm trying to relate it, biological process to oil industry. So, but before jumping directly to the application, we need to go back and say about the production, and then the purification, the chemical structure, identification, and then we will finish with the application by itself. So, as you can see here, production. The group started doing screening about potential biosurfactant producers. And we found several yeasts, bacteria, even fungus. But we find a really special one. So basically we are talking here about a wide strain, which is a bacteria, Bacillus subtilis, and it was named LB5A. So that time we have been using just this bacteria as a biosurfactant producer. So this is the bacteria. And when you related that bacteria with, to the literature, we found that probably that microorganism produce surfactin, which is a lipopeptide. Basically, if you have a look on the structure here, we got 7 amino acids, which is linked to a fatty acid. So basically, it's an amphiphilic compound. That means that compound has a hydrophobic and also a hydrophilic parts. So basically, it's a soap, a detergent. But if you think about surfactin, that structure is not well defined. That means depends of the microorganism that amino acids would change, or culture medium, or even fermentation parameters. And also the fatty acid length, from 13 to 16, more or less. So actually it's a mixture. With the literature defines as a surfactin is actually a mixture of structures. So we got a strain, biosurfactant producer, and then now we started to think about the culture medium. This is a really common approach on research, trying to use wastes as a culture medium because it's free, it's cheap. And you have been trying to use waste from corn industry, grape, wine, and potato, every kind. But we found the best one is from cassava industry. In particular from flour cassava industry. This is basically an illustration about the factory. So basically the roots came here and is peeled off, and then it cut it, and the next step we press that as a drying process. When you press that, a liquid comes out of that, right? And this is the waste. This is the cassava waste water. The remaining powder goes to the next stage and so on. So here we collect our culture medium, basically. I think in Europe it's not common cassava. So I'll introduce you cassava. We can grow in Brazil, or even I think in Africa or warm countries during the whole year. That guarantee me a continuous supply of culture medium during the whole year doesn't matter if it's winter or summer, doesn't matter. We got. And they have been testing that culture medium with different kind of microorganisms. And for yeast is really good culture medium, bacteria as well, but why? So if you have a look on the composition of the culture medium, we can see we got calcium, magnesium, copper, different sources of nitrogen, and also sugars. So you got everything in that culture medium. So at that point, we got a producer, a microorganism, we got a culture medium. We started to move on in different strategies, production strategies. So it did that in batch fermentation, in feed batch fermentation, and also a continuous fermentation at different scales. For instance, flex fermentation, Erlenmeyer's, top bench bioreactor, 7 liters, more or less 3 liters working volume, and also a pilot scale, 8 liters. And recently, more or less in 2011, we started that approach with simultaneous production. That means we can produce surfactin and also other biocompounds in the same fermentation. And here is some examples. For instance, this is a kind of flavor. We got just a few minutes ago a talk about it. And also enzymes. I'm going to show you how to get the whole pack of products. So basically, as I said, surfactin is a kind of soap or detergent. We can picture that. And if you think about a bioreactor, we got aeration there and also agitation. When you do that with detergent, that makes foam, right? We can collect that foam from the top of the bioreactor. And that's going to happen here, as you can see. The enzymes is still in the culture medium, so we can get the culture medium and get the enzymes. And also, if you separate it here, this part, we can get the flavors, or bioflavors, actually. So let me point something out here. We have been doing analysis on this flavor with SPME and also with liquid extraction. This is a kind of unique approach, right? So about the production, we have some papers, as you can see here. Production of biosurfactant using cassava wastewater as a substrate, production and stability of Bacillus subtilis biosurfactant, and so on. And recently, we have been working on the simultaneous production, as I said. So, and about purification? Because purification should be the next step, right? First of all, we produce that, and then we purify it. So basically here, my work started on the purification step. So back in Brazil, no one knew it, how to purify surfactin. And basically around 2008 to 2009, really nice papers came out from England, from a research named Dr. Jauregui. And I read her paper, and I was kind of stunned. Really nice results, really nice techniques. She didn't make up that approach to purify the surfactant, but she did improve it a lot. And we started to talk, and she accepted me for a split PhD, so she is my third supervisor. And we carried out a project named ultrafiltration in 2 steps of surfactant produced by Bacillus subtilis using cassava wastewater as a substrate. I'm going to show you how we purify that, but before I have to mention something here. So basically, picture we got a glass water, right? If you measure the surface tension of the water, the pure water, we got something around 72 millinewtons by meter, right? Something like this dot, red dot. So if you take a drop of detergent or something like this, a soap or a biosurfactant, and put in that water, a surface tension will decrease, of course, in this point. If you keep going, that means increasing the concentration of surfactant. At certain point, a self-structure will be formed. That point, or that concentration, is named critical micelle concentration, or CMC. And I want to point out or highlight that difference between synthetic surfactants and biosurfactants. For instance, surfactant has a CMC around 10 milligrams per liter, which is really close to hamnolipidus, which is another biosurfactant. When you have a look on synthetic surfactant, that's really higher. And as a general rule, the lower CMC, the more powerful is the biosurfactant, right? So basically, this is the approach about the purification. We produced that, we got a higher concentration 10 CMC, so we got self-aggregation forms here, and we got a lot of impurities, protein, amino acids, salts, ions, so a lot of impurities. So basically, we can carry out ultrafiltration process, the first step, and basically the low molecular weight compounds will permeate it, right? But the larger one, like micelles, will be stayed as well, proteins and everything. After that point, if we add any organic solvent, at this point, basically this kind of structure will disrupt it. And we got something here. And if we carry out a second step of ultrafiltration, we got our surfactant here, and the proteins would be retainted. Personally speaking, I think isn't a complex way to purify. It's a membrane-based technique, and that's great. That's make great and feasible to scale up or everything. And I'm really glad to say, yesterday was talked about as a research if we should go for patents or not. I'm glad to say my opinion here or express my opinion. I think we should go for a patent. And the last week, we got a patent about the production and the purification about surfactant. And all right, so we went to production, purification, and now we can identify the chemical structure of surfactant. And for that, we have using basically both two techniques. One is NMR, really nice technique, and the other one is Molotov MS. So basically, the Molotov MS data says that we have 11 surfactant forms. That means I can have there, among these 11 isoforms, different length waves or even different sequences of amino acids. Although NMR data showed that we got two sequences of amino acids. So for we remember how is the structure, we got 7 amino acids here and 1 fatty acid. And this is the result of isoforms that we got. Looks like the same, but they are different in 2 amino acids. And that thing really intrigues me. I was wondering myself many times if you, first of all, if it's possible separated each isoform, and if each isoform has different properties. That's really nice, but I'm not sure if it's possible to do that separated each isoform. So basically, we produce it, we purify it, we identify that, and now we can start to think about the application. If you think about reviews of biosurfactant in general, there are plenty of application or potential application. Since antimicrobial, emulsifier, food, cosmetic. I don't know, after a lot of work on the production and with the purification process, I think you have to really careful think about how to apply that compound. And I think the best way, or which is the best potential, is a technique named microbial enhanced oil recovery. And that's related with the oil industry. So basically, when you face it a well, oil well, that system is under pressure, high temperature, and even at high ionic strength. So when you reach that well, the oil comes out. Like this is like the picture of the oil recovery, right? After that, the second recovery is you have to pump in water or stream and pump out oil. Basically, the oil industry stopped here, the oil recovery, because after that it's so expensive or so hard to get oil out of the well. Thus, the aim of the microbial enhanced oil recovery technology is mainly the remaining oil in the wells after the primary and secondary techniques, which is 3 trillion barrels. So basically, if we think about all already explored oil, wells, there is oil in there. In each well, there's a lot of oil. And this is about 3 trillion barrels, right? And how the surfactant can help us with that? Basically, after the second strategy to recover the oil, we got something like this: rocks and crude oil and the base. And if you apply biosurfactant there, we can much more easily get the oil, right? Because if you put the biosurfactant, the surface tension will be reduced, so it's easier to get the oil out. And this is a group in China which has been working with that kind of approach. And they describe it that in Dakin oil field, the temperature ranged from 45 to 89 degrees Celsius, and the ionic strength is about that number. That is, the interaction of this parameter has to be considered. My point is, if you want to apply biosurfactant there, we have to guarantee that our biosurfactant will be stable at these conditions. So that's basically the main reason why not to use synthetic surfactants, because synthetic surfactants will not get this condition, will break. So basically this study described for the first time the surface activity measurements of surfactant at extremely condition of temperature, ionic strength, and pH, one at a time, and its interaction. So basically one at a time, we got pH here, we got ionic strength and temperature. As you can see in temperature, the surface tension is still stable. That means temperature is not affect the surface tension of surfactant. Different than ionic strength. If you have a look here, we are increasing the concentration, the ionic strength, and the surface tension is increasing as well. And pH is a kind of extremely pHs would be influenced here, right? So as a conclusion, we can say that temperature does not affect the surface activity of surfactant. So the next step is try to evaluate how this parameter works together. And for this, we carried out CCD, or Central Composite Design. As you can see, we carried out 17 experiments. For instance, the first one, we got this temperature, this pH, this ionic strength. The second one, we got the same pH, the same ionic strength, but the temperature is different. So that was the approach. As a result, it's similar to the experimental one at a time, because the temperature really doesn't not affect the surface activity. We did the math, and we got this equation. And basically, this is the most important thing to say here. Indicates that 18, about 90 grams per liter of brine or ionic strength is the threshold when associated to extreme pH and temperature. So basically, if you think about apply surfactant in the well, we can forget about the temperature or pH. We need to focus only on the ionic strength or the brine. Okay, we got a production, we got a purification process, we got a potential application since the surfactant is stable at extremely condition. Now we did test, we tested which oil should be better to recover it. So we got heavy oil, medium oil, and light oil. This is named oil displacement test. Basically, the higher area here, the good result is it, right? And we did that for standard surfactant and also for the surfactant which we produced. And the results indicates that the best short of oil is the heavy oil. So basically, in conclusion, the surfactant that was produced by cassava wastewater as a culture medium and purified by ultrafiltration has remarkable surface activity under extremely condition and also great potential to be used for microbial enhanced oil recovery. I would say thank you for my sponsor. Actually, also for my group in Brazil, my supervisor, my colleagues, my supervisor in England, Dr. Andrade. This is my email, and danke schön.
If you would like to patent your method, then publish. Because if you mention your non-publication, it's unpatentable.
Yeah, yeah.
And the question is, surfactant is only 5 single amino acids.
Yeah.
Did you try to synthesize it?
No, I think it should be really hard to synthesize because that's the point. I don't know how to synthesize that thing. For instance, this is a cycle of amino acids. Some amino acids is a D configuration, others are L configuration, and how to link that thing to, how to link the fatty acid to the cycle. I think it's easier to do the fermentation process, right? And this is the reason that biotechnology process are really really important when you compare to synthetic chemical compounds, right? Yeah.
Now actually there's many companies that it's easier to synthesize small peptides.
And how to link that thing.
This is chemical
Yeah.
structure.
If you, I don't know, if you think about how the microorganism build that structure, I don't know, that would be easy. Just react to something and get the surfactant. I think it would be tough, really, really tough. And about the publication, yes. Now we got the patent. I hope in the next months or weeks we came out with really good papers. That's my hope. You know?
Do you measure the molecular weight of this biosurfactant?
Yeah, actually, these Molotov result data, they show it not the weight, but the reason weight charge, right? His approach is around 1,036 Daltons, kilodaltons, but yeah, but that.
The molecular weight is changed when you change the culture condition or the medium.
Yeah, actually the literature says no. No, doesn't matter if you change the culture medium or the fermentation parameters, but personally speaking, I believe, of course it's change. Of course it's change. But
I would like to know, I have two questions. If you want to pump your product into a hole, basically, why don't you purify it at all?
for instance, because when you produce that thing, of course the microorganisms produce also protease. And if you take the surfactin, which is a lipopeptide, and protease, for instance, the protease can act on the surfactin. And also, I think if it's possible purify, you can control much better a system. For instance, each well has a different composition of oil. So if you mix it, that different composition with a product which has a lot impurities, how can I find a pattern? Would be really, really hard. And let me get back here. And my proposal, my aim, is basically produce surfactant and then apply that. So that's named ex situ application, right? But this group is trying to doing an in situ application. That means they want to grow the microorganism and then inject that in the well. But I don't know. If you think about it, it's a fermentation process in the well. And there's no agitation, there's no aeration. Also, the microorganism will produce acids, protein, I don't know. And as a conclusion, for instance, this paper is not reproducible. Of course, it's like a fermentation in the well, oil well.
But in any case, if your well is already pretty empty, basically.
No, that's about 50%. It isn't empty.
In any case, you really have to think about the costs here.
Yeah.
And if you can cut the purification step, that's enormous amount of cost.
Yeah.
And also, how do you, I mean, if you really want to apply this in basically every oil field there is.
Yeah, that's the point.
I have no idea how much of your wastewater there actually is available.
Yeah, definitely. Yeah, definitely. It's a good question. I would think, for instance, we are doing research, right? I think you have to keep going with the purified surfactant to try to find the trends and patterns and everything. After that, of course, we can think about it and skip it, the purification process. This is, of course. But for this moment, I think is the best idea is keep going with the purified surfactant. And about the culture medium, we can replace that, or I don't know.
And my last question would be, but that was probably not really focused
Yeah, you have to think about it is a kind of emulsification process, right? So the emulsification process has a time. For instance, it's not a stable system; it's a dynamic system. For instance, if you do if you carry out emulsification process in a test tube, for instance, and wait 1 week, 2 weeks, that is gonna separate, because the emulsification is not stable for too much time, right?
Currently, I think the water which is pumped into the well is being reused as mineral water, so because the separation is almost 100%.
Yeah.
But as soon as you use surfactants, the separation, even if it...
Yeah, that I don't understand. It could be a problem for the next step for the oil refine, but otherwise we got oil. So this is other matter, further matter.